Hi everyone, my name is Julia Cao. I'm a science tutor at the Science Hotspot at Lone Star College University Park. For my presentation today, I will be covering Biology 1. This is meant to be an ultimate survival guide for those who are taking Bio 1406 or 1408. So let's go ahead and take a look at some of the topics that I will be going over. So, on the right side, these are some of the topics that I will be covering today. And at the end, I have some advice and tips for those who would like to see how they can succeed in biology one. Sorry. Before I go ahead, though, I would like to clarify that I have tried my best to include as many topics as possible, especially those that are covered in both BIOL 1406 and 1408. However, there may be one or two topics or terminology that is not typically taught in biology 1408. For even further clarification, 1406 is biology for science majors, while 1408 is biology for non science majors. I have personally taken biology 1406, which is for science majors, so I am more familiar, or I am pretty familiarized with the topics here. But perhaps for a student who is taking biology 1408, there may be one or two types of concepts or terms that is not usually taught in the class. But once again, I have tried my best to include topics that are seen in both 1406 and 1408. So let's go ahead and take a look at our first topic. So we will be going over the pH scale, acids and bases. So a pH scale is used to measure the acidity or basicity of a solution. On the right side here, I've included an example of the pH scale. And the scale here has numbers which will range from 0 to 14. An acid is a substance that contains hydrogen ions. The more hydrogen ions there are, the more acidic something will be, and it will have a lower pH number which ranges from 0 to 6. A base, also known as alkaline, is a substance that has hydroxide ions. The more hydroxide ions there are, the more basic something will be, and it will have a higher pH number which ranges from 8 to 14. So here I have some additional terms. So if a solution is neutral, this means that it is neither acidic nor basic, and it will have a pH number of 7, which over at the scale over here, it is at the middle. A buffer is a substance that resists changes in pH, and it helps keep hydrogen ions relatively constant. So if something is becoming a little too acidic or a little too basic, a buffer will help control those changes and keep the pH level stable. So here in my notes, I've included that the lower the pH, the more acidic a solution is. The higher the pH, the more basic a solution is. Each unit on the pH scale represents a tenfold change. For every pH number that increases or decreases by 1, there's an exponential increase by 10. So what this means is if I were to compare between a pH of 6 versus a pH of 5, there is a change of pH number, which is 1. But when I'm putting it in a statement, I would not say that a pH of 5 is 1 times more acidic than a pH of 6. And that is because there's a tenfold change in terms of acidity and basicity. So in a statement, I would say instead, a pH of 5 is 10 times more acidic than a pH of 6. And I got the 10 from right here because it changes by a pH of 1. But when we're talking in terms of how acidic or basic something is, it changes by a multiple of 10. And this number exponentially adds up. So we'll go from, if you jump twice from, let's say, a pH of 6 to 4, it would increase by 100. And then you could say if you jump 3 times here, then it would increase by 1,000, and so on. And over on the top right, I've also included another example here. This time it is just going upwards. And at the arrows on the sides here, again, the higher the pH number, the more basic something will be. And the lower the pH number, the more acidic something will be. Let's go ahead and take a look at some examples on the next slide. So I'm going to go ahead and go over the first example, but if you would like to see the other 2 examples I have below here, feel free to pause the video here. So let's go and see example 1. It is comparing a pH of 1 versus a pH of 5. So yes, so on the scale here, 1 would be right here, and then 5 would be right here. Please ignore this area here, the tens, the jumps that I've included on the side. I just forgot to take those out. But if we go ahead and compare these 2 numbers, it takes 1, 2, 3, and 4 jumps to get between 1 to 5. Now, if we were to compare how acidic or basic, how much more acidic or basic these numbers are, we would have to figure out how many times it exponentially increases by 10. So from 1 jump is times 10. The second jump is times 10. Again, please ignore the numbers in black here. I apologize for not taking those out earlier. 3 jumps is another 10, and then lastly, we'll include times 10 here for one more jump right here. So it's 10 to the power of 4, or if we want to say the longer way, it's 10 times 10 times 10 times 10, and the product of that is 10,000. So if we were to put this in a statement, if we were to try and see how much more acidic pH of 1 is, we would say a pH of 1 is 10,000 times more acidic than a pH of 5. If we were talking in terms of how much more basic something is, then we would say a pH of 5 is 10,000 times more basic than a pH of 1. So again, I've included some more examples here. Feel free to pause the video if you would like to work on those examples or take a look at the pH scale on the side here. So our next topic is macromolecules. Here's just a general overview of macromolecules. So there are 4 types. We have carbohydrates, lipids, proteins, and nucleic acids. In terms of elements, all 4 macromolecules have carbon, hydrogen, and oxygen. If we take a look at proteins and nucleic acids, they both have nitrogen, and only nucleic acids have yet another additional element, which is phosphorus. If we take a look at the monomers here, it is simply just a building block of a macromolecule. So everything listed under this column shows what each macromolecule is made of. So for carbohydrates, it's monosaccharides. Lipids is made of fatty acids and glycerol. Proteins are made of amino acids, and nucleic acids are made of nucleotides. For functions, carbohydrates mainly provide energy. Lipids help store energy in fats and membranes. Proteins serve as catalysts, defense, transport, support, regulation, motion, and storage. Lastly, nucleic acids help store and transport genetic info. Our last column here shows how many kilocalories there are per gram. Carbohydrates have 4, lipids have 9, proteins have 4, and nucleic acid have 0. If you would like to take take a look at this table, feel free to pause the video here. So for the next couple slides, I have some quick facts for each type of macromolecule. So let's go ahead and start with carbohydrates. Again, monosaccharides are monomers or building blocks of carbohydrates. They are single sugars, and some examples include fructose, glucose, and galactose. Next, we have disaccharides, which are double sugars. Some examples include sucrose, lactose, and maltose. Lastly, polysaccharides are simply long chains of monosaccharides, and they have more than two sugars. And some examples include glycogen, cellulose, and starch. In the notes section here, it says the ending root word "ose" is seen in the names of many sugars and therefore generally associated with carbohydrates. In the future, if you come across a word that has this ending root word, it is most likely a sugar and it is most likely a carbohydrate or a type of example or type of a carbohydrate. So for lipids, triglycerides are one type that is made of one glycerol head and three fatty acid tails. And again, the building blocks or monomers of lipids are glycerol and fatty acids. Phospholipids are another type of lipids that is made of one glycerol, two fatty acid tails, and one phosphate head. So the heads and tails are in quotation marks, and on the right side here, um, this is a lipid or phospholipid. So here's the head, and here are the two tails here. In this section, it also says the tails are nonpolar and the head is polar. And in just a bit, I will get to what nonpolar and why the tails are nonpolar and why the head is polar. So phospholipids are generally seen in a structure called the phospholipid bilayer. So it's just simply two layers of phospholipids, and the word bilayer has the prefix bi in it, which means two. So it is two layers. So it also says here the polar heads point outward while the nonpolar tails point inward towards each other. So if we take a look here, I'm going to do purple. So the heads, if we take a look at this here and compare it to this picture here, the red portion is pointing outwards and the tails are towards the inside. So so we typically see this phospholipid bilayer in cells, generally animal cells, and we see it in the membrane of the cell, which is that outer covering. And don't worry about that. In a later slide, I'll go into depth about the cell. So in the notes section here, I've included that in this bilayer, the polar heads are hydrophilic and the nonpolar tails are hydrophobic. So the term hydrophilic means that it is attracted to water or water loving, and hydrophobic is that it is fearful of water, or it hates water. And in the picture here, it also says it right here. So the head loves water and it is therefore hydrophilic, and the tail hates water and it is hydrophobic. So for proteins, they mainly have four structures. The first one is called the primary structure, and it involves a chain-like sequence of amino acids, which, if we remember, amino acids are monomers or building blocks of proteins. Next, the secondary structure involves amino acids folding into either a flat or pleated-like sheet or a helix. A tertiary structure involves a globular protein which folds into a final 3D shape. And then a quaternary structure involves the protein being packed together with more than one amino acid chain. So if we take a look at the picture here, the primary structure is it looks like a chain. And then with the secondary structure, it can fold into either a pleated like sheet like this, or it can fold into a helical shape here. Like it looks like a squiggly line here. With the tertiary structure, the protein is almost It's not exactly globe like a sphere, but it's kind of like clumped together, and it's in the process of folding into its 3D shape. And then with the quaternary structure, it involves proteins being clumped and packed together here. And the example that's being provided here is with hemoglobin. So in my notes section here, I've put that proteins can be unfolded or damaged by a process called denaturation or denaturation. This is influenced by the protein being exposed to pH, to temperature, or to a solution's ionic concentration. For the last quick fact I have here, it is for nucleic acids. So, if we remember back at the overview, nucleic acids are made of nucleotides. Some examples of nucleic acids include DNA and RNA. So, DNA stands for deoxyribonucleic acids, while RNA stands for ribonucleic acids. So, with DNA, it is a double helix strand that is located in the nucleus, and it uses the bases A, T, C, and G. With RNA, it is a single strand that is located in the nucleus and cytoplasm, and it uses the bases A, U, C, and G. So, with these bases, they're just short for adenine or adenine. On this side here, thiamine for T, C for cytosine, and then G for guanine. T is typically seen with DNA, while U, which stands for uracil, is seen with RNA here. So for my notes here, I've put that there are base pair rules for both types of strands. So within DNA, A or adenine will pair with T, thymine, while for RNA, adenine will pair with U, uracil. For both DNA and RNA, C, cytosine will pair with G, guanine. So our next topic will be going over the parts of the animal and plant cell. Here's just a general overview of these types of cells. The top one, it shows an animal cell, while the bottom one shows a plant cell. So in my notes here, I've included animals and plant cells are both eukaryotes, which means that they both have a nucleus. However, these two cells differ in certain organelles that are specialized for either one of the cell. This section will go over the important organelles present in both cell types, as well as those that are seen only in an animal or plant cell. So to start off, we have here the nucleus, which is right here. Or let me do a different color so that way it can stand out. Let's do it in red, or no, let's do it in pink. So the nucleus is right here. Let me clear that. Or here. Yeah, let me reach all that way. We can keep track. So the nucleus is where DNA is stored, and it manages all cell functions in eukaryotes. The cytoplasm is right here. Or, well, the cytoplasm, as we'll see in the description, it is generally the space around here. But for the definition I've included here, it is simply a semi-fluid matrix located in the cell. And it is located outside of the nucleus. This is where all organelles are located, and it contains a fluid called cytosol. So the cytoplasm, again, is essentially just this entire space in the cell right here. So it would be here. So let's see. Let's do a different color. So the endoplasmic reticulum, or the ER, can be either smooth or rough. So here we have the rough. Oh, Okay, that covered the rough, but yes, this is one of the types of ERs, and then down here we also have the smooth endoplasmic reticulum. So with the rough ER, which is up above, it is mainly involved with protein modification, while the smooth ER is involved with lipid synthesis. With the ribosome, which is located here, as well as at the bottom here, this is where proteins are synthesized. And we can find these ribosomes on the rough ER as small bumps here, which I'll draw little dots here to represent those, or they're also floating freely in the cytoplasm, which where it says free ribosome, it's just essentially located in the cytoplasm, which is this yellow area I've marked here. Lastly, we have the nucleolus. I'll mark that in purple. So it will be right underneath the word nucleus right here. This is just essentially where ribosomal RNA is synthesized. So the nucleus is the bigger sphere, and then it has a little structure on the inside, which is the nucleolus. So here's just a continuation of other organelles in these cells. So the Golgi apparatus is located in this image. It is right here. It is also called the Golgi body. This is also known as the cell's post office, and this is where proteins are packaged for transportation around, inside, or outside the cell. The mitochondria, which should be right here, mitochondrion, it is also known as the powerhouse of the cell. So this is where it produces a usable form of energy known as ATP, and it has its own form of DNA. We also have the plasma membrane, which is right here, in which, if we remember back in the macromolecules section, it is the outside part of the cell. So here I've put that it surrounds the entire cell, and it controls what enters and leaves the cell. And the reason why is because it has that phospholipid bilayer. So in the notes section here, I've included that the plasma membrane is described to be selectively permeable, or its barrier is selectively permeable. So what this means is that it only allows some substances to pass into or out of the cell. The cytoplasm is, which is this yellow part here. I'll go ahead and redraw that. It's right here, and then it's this entire space here. This was it. The cytoplasm is also known as the intracellular matrix, and if there is any fluid that is located outside the cell, so essentially it is all this white area here. If we imagine it as a fluid space, this is outside the cell, and it is called the extracellular matrix. So lastly, this is the last parts of, oh, sorry, this is the last couple organelles that we have here that are seen in both types of cells. So we have here the vacuole, which I believe, oh, okay. There in the bottom picture here, there isn't any vacuole, but it is known as the cell's storage center, and this is where in plant cells it helps maintain water balance, while in animal cells it helps maintain waste products. And that's a typo there. Sorry, I meant to put products instead of produce. With lysosomes, here we go. There's a example here. So the lysosome is right here. So this is also known as the cell's waste disposal. So it digests and destroys excess cell parts, food particles, and invasive substances such as bacteria and viruses. The cytoskeleton here, we I can't see it on this picture here. It is just a network of protein strands that provide cell structure. So I'll go ahead and try and draw like an example. It's just like a whole network of strands that are all over the cell. It's kind of like a spider web almost here. And this cytoskeleton helps keeps organelles in fixed locations, so it prevents it from moving around like too much. Lastly, the flagella, which is the image on the top right, is a long whip-like structure used for movement, and we see this in human sperm cells. So here we have other organelles, but these are quite unique because they may either be seen in a plant cell only or only in an animal cell. So the first organelle we have is the chloroplast. So this is only seen in plant cells. It contains a substance called chlorophyll. And this chlorophyll is just a pigment that is used in photosynthesis to make glucose. And it has its own DNA similarly to the mitochondria. The next term we have here is the cell wall, which is also only seen in plant cells. It is a protective structure that supports the cell's shape, and it prevents excessive loss of water in plants. So since the cell wall is only seen in plant cells, animal cells have that plasma membrane. Our last term here is the centriole, which is mostly seen in animal cells. It helps with cell division, and I say mostly because it is seen in all animal cells, but it is rarely seen in plant cells. So on the right side here, I have a plant cell here, and as you can see, it has all the other organelles that was previously mentioned, and it includes the vacuole here, which I'll go ahead and label. It is that big storage center that's right here. And then, yeah, and then the chloroplast is over here as well. I'll do a different color so it's easier to see. It's that green structure here. And yeah, that's everything. Oh, here, one more. We have the cell wall too. So it's just like this in this structure around the cell. The next topic we'll go over is passive versus active transport. So what exactly is passive and active transport? Passive transport does not require ATP, and it moves from a high to low concentration. You could also say it goes down a gradient. Active transport, on the other hand, does require ATP, and it moves from low to high concentration, or you could also say it goes up a gradient. I like to think of a gradient as like a waterfall, and I like to think of fish swimming in that waterfall. So in terms of passive transport, the fish are going down that waterfall, or realistically, they usually go swim in a river, but they can also like jump. I think they can swim back up like against the river's currents. But so let's think of a gradient as a river instead. I think of a fish going down that river, and I think of passive transport. And in terms of active transport, I think of that fish like jumping up um, that river and going against the current. So, in terms of passive transport, which again does not require ATP, there are three types. The first one is simple diffusion, where molecules move from high to low concentration, and the purpose of this is to achieve equal concentration in all regions. With facilitated diffusion, molecules use proteins to move from high to low concentration. So, two types of proteins include channels and carriers. Protein channels allow passage of ions, while protein carriers help transport ions in solutes. The last type of passive transport is osmosis, where water moves from high to low concentration, and it can be hypertonic, hypotonic, or isotonic. So in terms of osmosis, there are three types. So with when a solution is isotonic, that means it has the same amount of solute concentration both inside and outside the cell. So isotonic is right here in this image here. With hypertonic, if a solution is hypertonic, that means it has more concentration or more solute concentration outside of the cell, which hypertonic is the left side here. And because of this, if a cell is placed in a hypertonic solution, it may shrink because they lose water. In terms of a hypotonic solution, it has more solute concentration inside of the cell. So hypotonic is here. Now, if a cell was placed in a hypotonic solution, it may swell up and burst because they gain water. In the notes here, I've included that during osmosis, water will follow wherever the solute concentration goes. Remember, it goes from high to low levels of concentration. So if a cell was placed in a solution, the water that's there will go wherever there is more concentration of solute. So it'll just follow wherever, and it will affect the shape of the cell. Which if the solution is hypertonic, that cell will shrink, and if it is hypotonic, that cell will swell up and burst. With active transport, which does require ATP, there are three types. The first type is protein pumps, which help move ions across the cell membrane from low to high concentration. And one example includes the sodium potassium pump. Coupled transport occurs when two substances are transported across the cell membrane simultaneously from low to high concentration. With bulk transport, large substances are transported without going through the cell membrane from low to high concentration. And some examples include endocytosis and exocytosis. So, for bulk transport, endocytosis is the movement of substances into the cell. And some examples include phagocytosis, pinocytosis, and receptor mediated endocytosis. With an exocytosis, this is the movement of substances out of the cell. So, endocytosis is into the cell, and then exocytosis is out. And with this image here, I'll go ahead and draw. This looks a lot, this is pretty like loaded information, but essentially exocytosis is right here, and then endocytosis is right here. So don't worry about the other terms that are here. With my notes here, I've included that the prefix endo means from within. The prefix exo means from outside. So the letters en in endo is similar to the word enter, and the letters ex in exo is similar to exit. So endo could be, you could think of something entering the cell, and then for exo, you could think of something exiting the cell. So with endocytosis, there are three types of endocytosis. So the first one is phagocytosis, where the cell takes in solid matter, and you can think of it as cell eating. With pinocytosis, the cell takes in liquid fluids, and you can think of it as the cell drinking. Lastly, receptor-mediated endocytosis is where the cell takes in molecules when they're bound to a receptor on the membrane. So phagocytosis is here, and it takes in solid matter or large particle. With penocytosis, here it takes in liquid fluids, and then receptor mediated endocytosis has these little receptors here, or sorry, not the ligand, sorry, the receptor right here. The ligand is what binds to the receptor. For my notes here, I've put that the prefix phago means to eat, and then pino means to drink. So eating and drinking. Our next topic is mitosis versus meiosis. So, what is exactly mitosis and meiosis? With mitosis, it creates two diploid cells that are genetically identical, and this is represented by the 2N. This is also known as cell division for somatic cells or body cells, and it will replicate once in one stage. With meiosis, it creates four haploid cells that are genetically different, and it is represented by N. This is also known as cell division for gamete cells or also sex cells. It will replicate twice in two separate stages called meiosis 1 and meiosis 2. So if we take a look at the image here, mitosis is the stage that is above, while meiosis is the stage located below. So with mitosis, there is two offspring, and with meiosis, there is four. And again, mitosis replicates just once, and meiosis will replicate twice in two mini stages, with the first being meiosis 1 and then the second being meiosis 2. With the notes here, I've put that the processes of mitosis and meiosis are the same since they both go through certain stages. Those stages are prophase, metaphase, anaphase, telophase, and cytokinesis. The difference is that meiosis goes through these stages twice. And we will learn more about this in the cell cycle section. So here are some additional terms. So the first one is diploid, which is the 2N here, which we saw in the previous slide. So this just refers to two sets of chromosomes. This is seen in humans, in which we have 46 chromosomes. Or in terms of diploid, we have 23 pairs of chromosomes. In total, that would be 46. Half of the chromosomes come from each parent, so each parent has 23. Another term here is haploid, where it refers to one set of chromosomes, or represented by the letter N. This is seen in egg or sperm cells, in which it has 23 chromosomes. The note here says that the root word di in diploid means two. The letters ha in haploid is similar to the word half. So therefore, we could say that a haploid is therefore we could say a haploid is one half of a diploid. So what is a karyotype? A karyotype is a diagram that shows a visual appearance of an organism's chromosomes. Humans have, as previously mentioned, humans have 23 pairs of chromosomes, or 46 in total. The autosomes are also known as the first 22 pairs of chromosomes on a karyotype. So from 1 to 22 here, and everything in between, those are autosomes. With sex chromosomes, they are related to the sex of the offspring, and it includes the X and Y chromosomes, which is the 23rd pair here. The next topic we'll go over is the cell cycle. So with the cell cycle, there are there are officially four, but I will include a fifth one here, just for emphasis on that stage. So the first phase is called G1, which is the primary phase for cell growth. The next phase is called synthesis, or S, and this is where DNA replication occurs. The next phase we have is G2, where cells prepare to divide. Then we have mitosis, which is where the cell divides. And cytokinesis is, it really depends on the image. Some people will include cytokinesis in the cycle. In this image that I have here, it's not included, but essentially it is an important stage. This is where the cytoplasm of the cell will start to divide, and it will split into two new cells. So in my notes section here, I've put that the G1, S, and G2 phase are collectively known as interphase. Cytokinesis is not a separate phase in the cell cycle, but it rather happens towards the end of mitosis in a stage called telophase, which we will go ahead and get into in just a bit. So here we have the specific stages of mitosis. So up here we have interphase here, and that is where G1, S, and G2 are happening. And then we get into prophase. So with this stage, this is where individual chromosomes become visible, and the centrioles, which if we remember in the organelles section, centrioles help with cell division. So they begin to move to the opposite sides of the cell to form a structure called the spindle apparatus. So I'll draw, or I'll highlight this in a different color. Let's do purple. So the centrioles here are going to be circled in purple right here, and then in prophase they are on the opposite sides of the cell. So the next stage we have is metaphase, which is where the chromosomes meet at the middle of the cell. So they are right here. I like to think of the M in metaphase as in middle. So yeah, metaphase for middle. The next stage we have is anaphase, where the sister chromatids separate and move away to opposite sides of the cell. So chromosomes can be dubbed or considered as sister chromatids, and essentially chromosomes have two strands linked together. And then when you separate them apart, they become sister chromatids. So in anaphase, they separate and move away to the opposite sides of the cell. So if we take a look at anaphase, they're starting to move away from each other. With telophase, the spindle apparatus starts to disappear, and there's a nuclear envelope, which is a part of the nucleus. It forms around each set of sister chromatids. So it will be right here and right there. Lastly, we have the cytokinesis stage, which again, it appears towards the end, around the same time as telophase, like towards the end. And this is where the cytoplasm divides and the cell is split into equal halves. So this process occurs differently in animal cells versus plant cells. So with animals, there is a cleavage furrow that forms. So if we take a look at cytokinesis here in the picture, essentially a cleavage furrow is just like a pinched area of the cell. So it is right here, right where it is pinched, that is called a cleavage furrow. With plant cells, there's a cell plate that forms, and it eventually becomes the cell wall. So if I go ahead and just draw like an example here, I'm gonna draw underneath here. So let's say this is a plant cell, and it's already in the process of cytokinesis. So I'm going to draw the nucleus here. There is like a specific plate here that forms here, right in the middle. I'm so sorry for my horrible drawing. This cell plate here eventually becomes the cell wall of the plant cell, which, if we remember, it's that hard protective structure of the cell. The next topic we have is genetic crossing and inheritance. So, what is a genotype, phenotype, and Punnett squares? So, with genotype, it is the total set of alleles that an organism has. And this is typically written in uppercase and lowercase letters. So, some examples I have here are just the letter S, and it can be written both. It involves two letters, and they can be both written uppercase. It can also be both written in lowercase, or it can do a combination of both, where there's just one uppercase and one lowercase letter. With phenotype, it describes an organism's physical appearance, and we would refer to this with traits, qualities, and colors. So, for example, we could say something is round, we could say it's wrinkly, or we could say it's white. With Punnett squares, it is used to show possible genotypes and phenotypes of genetic crosses. So, a monohybrid cross is used to study two variations of a single trait, and it has a phenotypic ratio of 3 to 1. A dihybrid cross is used to study two separate traits within a single cross, and it has a phenotypic ratio of 9:3:3:1. In my notes here, I put that mono means one and di means two. So, with the monohybrid cross, we're looking at a cross between two traits here. So AB times AB. And then with the dihybrid cross, we're analyzing a cross between two sets of traits. So it would be AABB times AABB. So, what does homozygous and heterozygous mean? Before we describe those, we have to know what an allele means first. So, an allele is an alternative form of a gene, and it can be dominant or recessive. And a homozygous allele refers to two of the same allele, in which here the letters can be capitalized with big A, or it can be lowercase with little a, little a. With a heterozygous allele, it refers to two different alleles. So the one letter can be uppercase with big A, and one letter can be lowercase with little a. So if we take a look at the picture here, at this with the first picture on the left, there are big B's. So this the offspring will all be homozygous. With the second picture on the right, there are two different types of alleles. We have ones that are big B, little b, and then we have another that's just both little b's. So the ones that are big B, little b is homozygous. I'm sorry, not homozygous, heterozygous, while the little b's are homozygous. So in my notes here, I put that in terms of genotype, a dominant allele is expressed in capitalized lettering. Meanwhile, a recessive allele is expressed in lowercase lettering. We also should look at what a pedigree is. So this is used to analyze and determine the probability of genetic disorders in offspring, and it looks like a simple family tree. So my note here says that the main difference between a karyotype and a pedigree is that a karyotype analyzes chromosomes within an individual. Meanwhile, a pedigree shows a pattern of inheritance within a family. In a pedigree, if a circle or square is half shaded in or has a blend of colors, which we can see here, and there's also a couple more here. So all these that are shaded, like blended in, this just indicates that the family member is a carrier of the genetic condition or disorder. So we'll be going over cell respiration versus photosynthesis. So before we go into that, we have to know what an heterotroph versus an autotroph is. So a heterotroph means that they eat organic compounds that are produced by other organisms, and they are also known as consumers. With autotrophs, they are able to produce their own food from raw materials or energy, such as through photosynthesis, and they are also known as producers. So most autotrophs are plants, algae, and bacteria. And heterotrophs include animals, fungi, and many bacteria. And in my notes here, I put that the prefix hetero means different or other, and the prefix auto means same or self. So heterotrophs may rely on other, quote unquote, other sources, while autotrophs rely on, quote unquote, themselves. So, to briefly go into cell respiration and photosynthesis, cell respiration is where glucose is broken down to produce an ATP for energy, and the formula is glucose plus water plus oxygen becomes carbon dioxide, water, and ATP. On the other hand, photosynthesis uses sunlight to power synthesis of organic molecules, and the formula is carbon dioxide plus water plus light becomes glucose plus water plus oxygen. My note here says that glucose goes into cell respiration to make carbon dioxide, and carbon dioxide goes into photosynthesis to make glucose. So essentially, the reactants of the equation here can become the products of another equation. So glucose turns into carbon dioxide, and then in photosynthesis, carbon dioxide becomes glucose. And also in the note, it says here cell respiration makes carbon dioxide for plants who make oxygen in photosynthesis for organisms such as humans to breathe and survive. So, the last thing I have here is just some advice and tips. So for this survival guide, I'm pretty sure many students are wondering how can they succeed in this class. So some things to know is that biology involves a lot of memorizing, and in order to succeed in this class and ace the tests and quizzes, you will need to study and memorize terms. So I would suggest using active recall when studying, such as using flashcards and practice problems. You could also look up root words and prefixes online. And I've included some examples here, such as mono, di, tri, hetero, and homo. You could also study and review according to your learning style. So, if you're a visual person like me, I always like to look at pictures and models online or in person to take a look at specific structures or important features. If you have an auditory learning style, it could help to listen to videos, watch videos on YouTube, or yeah, watch videos on YouTube helps a lot. And with reading and write, if you have a reading or writing learning style, then making a guide, writing down a guide for the chapters and units that you're learning helps a lot as well. And if you need tutoring, you can always come by the Science Hotspot located in the CSI building at Lone Star University Park. And my note here says that for spring 2023, which this video is being recorded, tutoring hours at the Science Hotspot are from 8 a.m. to 5 p.m. So I also have some websites that you could check out. So you could, the most popular one is Quizlet, where you can do tests with only, where I found, from my experience, I found that doing tests with just only flashcards helped me a lot with active recalling. I also used a website called Purpose Games, where they provide games and quizzes that involve labeling. So for lab exams, you'll be learning, you'll try to have to identify specific features of, say, plants and animal cells, and purpose games helped me a lot with that. And you can also make the games yourself too. And the last website I would like to recommend is called Save All, where you can make your own virtual flashcards for studying and self-quizzing, and that also helps with active recalling as well. So yeah, if you have reached the end of this video, I would like to thank you so much for watching, and yeah, have a nice day. Bye bye.